When you work on a group project, each person has his or her own talents and skills that add a particular value to the group's work. In the same way, the components of an eukaryotic cell has a specific job. All the parts of the cell work together to help the cell survive. Hi, and welcome back. In today's biology segment, I'm going to talk about eukaryotic cells.
Eukaryotic cell structure. Objectives: identify the structure and function of the parts of a typical eukaryotic cell. Explain the advantages of highly folded membranes in cells. Compare and contrast the structures of plant and animal cells. In the cell, the plasma membrane acts as a selective permeable membrane, as shown in the picture. The fluid mosaic model described the plasma membrane as a flexible boundary of a cell. However, plant cells, fungi, bacteria, and some protists have an additional boundary: the cell wall. The cell wall forms an inflexible barrier that protects the cell and gives it support. The picture shows a plant cell wall composed of a carbohydrate called cellulose. The cellulose forms a thick, though mesh of fibers. This fibrous cell wall is very porous and allows molecules to enter. Unlike the plasma membrane, it does not select which molecules can enter into the cell. The nucleus and cell control. The nucleus is the leader of the eukaryotic cell because it contains the directions to make proteins. Every part of the cell depends on proteins, so by containing the blueprint to make proteins, the nucleus controls the activity of the organelles. The master set of directions for making proteins is contained in chromatin, which are strands of the genetic material DNA. Nucleolus. Within the nucleus is a prominent organelle called the nucleolus, which makes ribosomes. Ribosomes are the sites where the cell produces proteins according to the directions of DNA. For proteins to be made, ribosomes must leave the nucleus and enter the cytoplasm, and the blueprints contained in DNA must be translated into RNA and sent to the cytoplasm. Cytoplasm is a clear gelatinous fluid inside a cell. Ribosomes and translate RNA are transported to the cytoplasm through the nuclear envelope, a structure that separates the nucleus from the cytoplasm. Nuclear envelope is a double membrane made up of two phospholipid bilayers. Actually, you can see it here in the photograph. This is the nuclear envelope, and it contains nuclear pores for substances to pass through. Ribosomes and translate RNA pass into the cytoplasm through these pores in the nuclear envelope. Organelles for assembling and transport of proteins. One particular organelle in an eukaryotic cell, the endoplasmic reticulum or ER, is the site of cellular chemical reactions. It is arranged in a series of highly folded membranes in the cytoplasm. A large amount of folded ER is available to do work in a small space. The ribosome's job is to make proteins. Each protein made in the rough ER has a particular function. It may become a protein that forms a part of the plasma membrane, a protein that is released from the cell, or a protein transported to other organelles. Ribosomes can also be found floating freely in the cytoplasm. They make proteins that perform tasks within the cytoplasm itself. Areas of the ER that are not studded with ribosomes are known as smooth endoplasmic reticulum. The smooth ER is involved in numerous biochemical activities, including the production and storage of lipids. The Golgi apparatus. After proteins are made, they are transferred to another organelle called the Golgi apparatus. It's a flattened stack of tubular membranes that modifies the proteins. The Golgi apparatus sorts proteins into packages and packs them into membrane-bound structures, called vesicles, to be sent to the appropriate destination, like mail being sorted at the post office. Vacuoles and storage. Cells have membrane-bound compartments called vacuoles for temporary storage of materials. A vacuole, like the one in the picture, is a sac used to store food, enzymes, and other materials needed by a cell. Animal cells usually do not contain vacuoles. If they do, the vacuoles are much smaller, as shown in picture B. Lysosomes and recycling. Lysosomes are organelles that contain digestive enzymes. They digest excess or worn-out organelles, food particles, and engulf viruses or bacteria. The membrane surrounding a lysosome prevents the digestive enzymes inside from destroying the cell. Lysosomes can fuse with vacuoles and dispense their enzymes into vacuole, digesting its contents. Chloroplasts and energy. Chloroplasts are cells organelles that capture light energy and convert it to chemical energy. Chloroplasts contain the green pigment chlorophyll. Chlorophyll traps light energy and gives leaves and stems their green color. Mitochondria and energy. Mitochondria are membrane-bound organelles in plant and animal cells that transform energy for the cell. This energy is then stored in the bonds of other molecules that cell organelles can access easily and quickly when energy is needed. The cytoskeleton. The cytoskeleton forms a framework for the cell, like the skeleton that forms a framework for your body. However, unlike your bones, the cytoskeleton is a constantly changing structure. Here you can see it in this photograph. It can be dismantled in one place and reassembled somewhere else in the cell, changing the cell's shape. And here you have a chart that compares both types of cells, prokaryotic cells and eukaryotic cells. So the plasma membrane, cell wall, and ribosomes are found in both, only that in eukaryotic cell is just present in plants. And the rest that we just saw in this presentation, chloroplast, cytoskeleton, endoplasmic reticulum, Golgi apparatus, lysosome, mitochondria, nucleus, and vacuole, are only present in most eukaryotic cells, and they are absent in prokaryotic cells.
I hope you found this video useful. Thanks for watching, and until the next one, bye!